Welcome today on the March 29th, 2025 to the University of Kentucky Aztec Lecture Theatre. And for those of you who've come here, thank you for coming. And those of you online, thank you also for participating. Today I'd like to talk about probably our most exciting research we've done to date, which is mitochondrial whole body transplant for eutherian mammals. I think this is sort of what we would like to call the holy grail of cellular science from our point of view anyway. So I'd also like to acknowledge our collaborators, Professor Tobin from the University of Kentucky and Mr. Decker from here at the Aztec Centre, as well as obviously Professor Champion down at the Mitochondrial Trust in New Zealand. So just a bit of background about the Mitochondrial Trust. This is Professor Liggins here in 1967. And that was the year of my birth. And the real significance here is that he changed the way that we all think about a fundamental thing in biology, which is childbirth. So until 67, everyone believed that childbirth was initiated by the woman in late gestation. And what he essentially showed was that that was wrong, that the fetus actually initiated both final maturation of its own self, so like lungs, surfactant production, all of that, and then initiation of parturition. So what that's meant in real life is the saving of millions and millions of lives since that. And I suppose we've tried to model our research together with the late Professor Liggins on actually having a very unique approach to biology. So in my 36 years, this would be the most exciting thing that we have done, as I mentioned. And essentially what we're doing is actually being able to now replace every single mitochondria into a patient. So replace all of the patient's mitochondria with brand new mitochondria from a donor, or from themselves, or from a what we call a homologous donor, which would be a close relative. So why would we want to do this? The literature shows that mitochondria leads to a lot of disease, and so transplant can lead to disease alleviation and also potentially to increase in longevity. So I just did this earlier today. This is just like 3 PubMed searches. And so if we put in mitochondria and disease, we've got 68,000 results. So that's how big this area of research is. We put in mitochondria and aging, we've got 17,500 results. And then mitochondria and longevity. So actually physically people talking about the excitement of this actually allowing eutherian mammals to age better, but also live longer, over 2,000 results. So just a bit of history on myself and mitochondria. So I started investigating mitochondria in 1990. This is an early 90s paper that I came out with that uses a stain called rhodamine, and this is scientifically interesting because rhodamine only fluoresces when you have a negative potential across the inner mitochondrial membrane. What that means in real time is that the mitochondria actually has to be functioning and has to be alive. So that's what this paper was all about in the early 90s. But you'll see in subsequent slides we have coming up, these are cells fluorescing with R123, the rhodamine dye, and that shows that the cells are alive. And you'll see the significance of this as we go on in the presentation. So we're going to go through the protocol, how we get there, but just to summarize it, we treat with chaperone proteins for a month to the patient, then we perform mitochondrial transplantation, which is intravenous, and then we follow this up with 3 months of chaperone protein treatment, and we'll go through the reasons for that. But first of all, we'll just talk a little bit about how fundamentally the cell ages and how that impacts on, or how the mitochondria impacts on the cell, and how chaperone proteins can help alleviate this in a temporary fashion. So just to get a schematic of this is the nucleus. We've got the Golgi here, but most specifically we're interested in the mitochondria and the cytoplasm. And so this is just mitochondria. And what happens is, as you either in a disease state or you're getting old, essentially, is your mitochondria start producing reactive oxygen species, which go into the cytoplasm, and they create havoc. And the havoc they create is basically on proteins. And what they do is they stop efficient protein production, and actually can create basically protein chunks that sit inside your cell and stop basic functioning of your cell. So that's the fundamental of all biological aging of cells. So you can see protein accumulates occur, which slows up the cell and cell functions. If you apply chaperone proteins, which is what we've been doing for a number of years now, we have the only commercial product available of that in the world, then those chaperone proteins will go inside the cell and they will go and clean up the protein accumulates. Clearly the problem with this is that your mitochondria are still producing reactive oxygen species, they're old, and so you're going to have to carry this process on. It's a pretty efficient process and we've had some great clinical results. However, the ultimate would be to be able to replace these little guys, and then we're not going to have the problem. So essentially what we're going to do, and this is, we're going to give some real life clinical example that what's happened is actually put mitochondria, for example, into the heart, into myocardial tissue, and replace the dysfunctional myocardiocytes with functional mitochondria to regain function. So not to bore with terminology, but a lot of the studies have been done both in lab animals and in humans with autologous, so taking mitochondria which are still operating okay in one part of the body and putting it into the part of the body where the mitochondria are not acting sufficiently. So we'll call this autologous. Homologous is using the same type of mitochondria, but it may not be from you. It could be a close family donor. And so, for example, a niece, for say down a female line, they'll have the exact same mitochondria. And so that's actually been used in an ovarian's experiment where women have successfully who've been infertile been transplanted with mitochondria from their niece and had a resultant baby because the new mitochondria have gone inside the oocyte, and then the oocytes have been able to be fertilized. The last one there is heterologous, and what heterologous is literally getting mitochondria from a different source. So from another individual that doesn't have the same parentage, or the same maternal parentage, and we believe we're the first people in the world that have done that. So myself, I received the first mitochondrial transplant in a eutherian mammal, a whole body transplant, and the donor was my son. So clearly that mitochondria is not from my lineage, from my female lineage, but from his mother's female lineage. So what the whole idea is with what's happened to date is mitochondrial transplantation happens in the organelle, so it's directly injected into the organelle. And the reason that whole body hasn't been seen as an option is that there hasn't been a way until now that the body can accept a whole mob of mitochondria. So I suppose the position that everyone's been in is how could we somehow isolate trillions of mitochondria to go into the body and somehow not have to inject every single organelle. So that's what we'll talk a little bit about today, about the hurdles that we've overcome. So this is a review of the entire literature. So mitochondrial transplantation has been shown in the rat model to help alleviate Parkinson's disease, in a cerebral ischemic model to bring back brain function as compared to the controls, and also to aid in diabetes. In the mouse model, again, it's been replicated to show to help alleviate Alzheimer's disease and also diabetic nephropathy. Sorry about that. So the other two species that have been looked at a fair amount is the pig. It's been shown that mitochondrial transplantation on a local level has been able to relieve renal ischemia, again in an ischemic model, and also relieve heart ischemia in a heart ischemic model. So just for those who don't really know what those models are, essentially you're cutting off the blood supply in a model and you're creating some apoptosis / necrosis of cells. So like the cells are just about in cell death or they have just died. And then you release the vasculature And so by the time you get reperfusion, you're going to have some cells that are already died, so they can't be brought back. You're going to have some cells that are in what we call a state of worse for wear, a great scientific word for that, coined by the Rolling Stones. So we know that if we give mitochondria to those individuals, then they will actually be able to respond and recover from the ischemic necrotic model. And so that's also been shown in a heart model in the rabbit. So just in the human, there's sort of just been 3 specific treatments. One I mentioned before, which was in infertility. Although the oocytes in suspended animation, the mitochondria is still functioning. So they are getting old, although all the genetic components and that are not getting old in the oocyte. So it's been shown to be a successful treatment for infertility in women who are in their mid to late 40s. So again, the donor could be from a niece, so we'd call that homologous, so it'd be exactly the same mitochondria. But what we're telling you today is that heterologous is also possible, that you could actually have a donor from another individual, could be a male individual. It's just mitochondria, because the mitochondria want to go inside. So these are direct models, been used with a group of children who FDA gave approval. They were all going to die of end-stage cardiomyopathy and all ended up living, which is just unbelievable, really. And also in acute respiratory distress. So just a real basic stuff about sort of mitochondrial function. So what we've been really interested in looking at before we started transplanting is how heritable are these mitochondria and what is the significance of this. So what we do is using things like the seahorse machine, look at real-time mitochondrial function. And so we can compare sort of older individuals with younger individuals and actually real-time measure that there is a significant decrease in mitochondrial function as you age, and also look at the inheritance of that. So this is using a horse model, and so basically what we found was that if you look at all these different parts of mitochondrial function in the respiratory model that I just put up there, that you have very significant comparisons between the mother and the foal. So obviously it doesn't matter if it's a male foal or a female foal. So the significance of that is that we know that with mares as they age, that the athletic ability of their progeny decreases. And I've often just believed it was sort of like that the placentas got older, that there's maybe fibrosis or some other thing going on inside the uterus. But it turns out that actually mitochondria is a key function of that, because let's just say that you did happen to get a 18-year-old mare pregnant, then her mitochondria that she's passing on are acting like an 18-year-old. They're not acting like a 6-year-old or an 8-year-old. So it's sort of interesting. If you look at international statistics, you'll often find that the most winningest progeny come out of mares that are under 8. So this shows that. And these are just very specific measurements of mitochondrial function. So it's not just a function of, okay, how much energy can the mitochondria produce, but how fast, for lack of better words, with oligomycin, how fast can you put the brakes on, and then how much recovery do you get. With maximal respiration, what happens if you just throw as much fuel at the mitochondria? Do they respond? And also, do they produce toxins? So that's very, very interesting stuff for us. And we know that this as a model is very, very sound for the human as well. So essentially what we're saying is that the inheritable traits of mitochondrial function do pass from generation to generation. I think there's a whole area of study that's about to open up in the world, and we're certainly pursuing that. And so essentially what we're saying is that younger mitochondria, they're not only more efficient, but they also produce less toxins. So there's less intracellular pollution, and that's sort of the key to anti-aging. So what were the hurdles that we sort of have had to look at for going from direct injection of mitochondria into an organ for incorporation to being able to do whole body? So first of all, you need a good source of donor mitochondria, and then you have to be able to process those mitochondria in a way in which you don't decrease the functionality. Because if you then go and transplant dysfunctioning mitochondria, I think personally that they won't take obviously we have never tried that experiment and won't be, but also if some of them did take, then that would be the worst thing. You might actually go backwards. So, so what you do in vitro with harvesting and processing is very, very important. And how can we then put enough mitochondria into the individual? And so what we came up with is an idea that if somehow we could embed mitochondria into the bone marrow and get it incorporated in pluripotent cells that are making white blood cells, then those new mitochondria will take over, and then as more and more white blood cells are made, then they will have the brand new mitochondria, and then they will go out to the whole body. And this is based on a lot of published science that essentially says that superior mitochondria want to take over cells with inferior mitochondria on them. So this is something that we talk about, we call it pruning. So it's literally like pruning a plant. There's a lot of biological examples of biological pruning. And this is on an intracellular basis. So what it essentially says is that the fittest will win, and then they will reproduce and they will take over. So we came up with a method that we call SWIP, which is separation of mitochondria without any impact physiologically. And so the whole key to this is that we have a process of mitochondria jettisoning, and so the mitochondria are literally thrown forward, and then they're caught like butterflies are caught in a butterfly net, so exactly the same. So therefore we don't have the problem with centrifugation and hurting their ability to function in their basic membrane. So that's where we were heading for, is a process to make 30 trillion new mitochondria. And so we knew that from using chaperone proteins that there was some type of unidirectional process happening, that somehow as you clean the cell, then the mitochondria wanted to go and stick to that cell. So what we found is that if you treat the chaperone proteins first, then the bone marrow, essentially, for lack of better words, is slightly cleaned up. And then when we add the donor mitochondria, they actually go directly to the bone marrow and get incorporated there. So they are being literally put in intravenously. So we got individual mitochondria in the bloodstream, and everyone knows that mitochondria need other organelles. So they have to go to safety, and we truly don't know how that happens. No one knows. No one even knows how mitochondria take over on direct injection on organelles. We just know that it happens. They must have some tropism towards cells, and somehow they must have some dominant ability to get into cells. So that's why you need to only give very, very good mitochondria. So this is just a little diagram. I mean, we literally just get blood from the donor, whether it be a horse or a human, and then that gets processed, and then we like to signal back to that original slide of rhodamine. I like to signal with a fluorescent probe, so then we know that those mitochondria are different than all the rest of the mitochondria in your body. So the millions that we're putting in are injected into the individual, and as I said, they can now be tracked. They can be tracked both intravenously and by intramuscular biopsy, and shown that they get incorporated. So this is actually this is pre-transplantation, so these are actually just mitochondria under bright field, and this the exact same field just with the fluorescent tag. So you can just see all those mitochondria are functioning. So the rhodamine is only fluorescent again if you have an intact inner mitochondrial membrane which has a negative potential, so that we know the Krebs cycle is working. So this is how it happens. The donor mitochondria get incorporated into the bone marrow, so then incorporated into pluripotent stem cells, which then go into various forms of white blood cells, and then they get distributed around the body. So this is actually an example of blood which has been pulled from recipient on day 3, and we find this over and over again. So far, we've only done 10 mitochondrial transplants to date, and all 10 have taken. Now, we don't know the long term of this. We'll be monitoring those individuals long term. The reason that we are doing this talk today is that it's been 1 year since the mitochondrial transplantation has been done, so we're making sure that all our clinical follow-up has been done on all individuals, both horse and human. And so, again, this is just this is looking at Brightfield, and that's just blood taken, and then you can see in a snapshot, this is just day 3 of mitochondrial incorporation into white blood cells. So going over the treatment again, we treat with chaperone proteins to clean the cells up, we do a mitochondrial transplant, and then we continue to use the chaperone proteins. If you have too many chaperone proteins, there's no untoward side effects, and your body will just get rid of them. It's just there to try to clean up the cytoplasm. So what I'm saying by that, if we think about that original slide with the junk in the cell, if you put a new mitochondria inside a cell directly, let's say, and you've still got junk in there, then you're still gonna have a problem because you're not able to make proteins. You have to clean the junk up with chaperone proteins, then you can put new mitochondria in. And we really believe some work that we've done is that without that cellular cleanup, the mitochondria actually don't have a tropism and don't know how to go into the cell. So just some exact results. In the horses, they've all been older horses, 20 to 27 years, all done by referring veterinarians that have a need to alleviate some aging issues. And so the aging issues have been lameness, a lot of infirmment, and acyclicity specifically in the mare, and some chronic wasting disease. So the clinical outcome to date in the horses is we have had lame horses become sound. I truly can't tell you the whole mechanism of action. It's sort of been astounding to me. Dramatic turnaround in physical condition, and also now we actually got a 27-year-old mare to actually start cycling again. So she'll actually be bred this breeding season, which will be very, very interesting to see if she takes. So the other thing that blindly people have noticed, their staff who didn't know anything about it have sort of said, hey, that horse is acting differently, acting in a more youthful manner. So horses they play a lot when they're younger, and as they get older, they probably get a bit like me, a bit crankier. But they literally in a paddock will be like mucking around and rearing and playing and joining. One particular case, the horse had been sort of shunned by the herd a bit, probably because it had ill thrift, but now totally accepted back into the herd, which is sort of interesting. So with the humans, they've ranged from 56 to 70 years, and the ailments that they had were alopecia, heart arrhythmia, arthritis, general ill thrift, and diabetes. And so the clinical outcome so far has been reversal of congenital heart arrhythmia of someone in their late 50s. Hair regrowth has been with all individuals, which has been really quite interesting. Exercise improvement, so these days with individuals exercising, they've got a really good idea of their heart rates when they do normal exercise, and also speed over that. So it's been roughly a 20% improvement, or it's an average of 20% improvement. With also a couple of the patients had a lot of arthritic issues, so there's been improvement. Arthritis hasn't gone away totally, there has been improvement. And one diabetic was treated, and they had already monitored their insulin production, their own natural insulin production, pre-treatment and post-treatment, and this is to 1 year again. It has increased 400%. So the other sort of miscellaneous things again that have come up is everyone who's had a transplant says that they've had an energy increase, an increase in libido, and memory enhancement, general vigor, and believe it or not, even muscle growth. So they've been the positive things to date. So I'd like to sort of tell everyone, if you've got more questions later on, please contact me. You'll see my email will be posted with this talk. And so I'd love to hear from you if you've got any questions or comments. Love to hear from you, love to know anything you can think about. So thank you very much, everyone, for their attention. And I'd just like to again acknowledge Professor Liggins, who started the research trust with me down in New Zealand. This is Professor Liggins with me a few years back, going to a big party at my brother's place. And also I'd like to acknowledge we've had our second member of my advisory board. It's not catchy, but Dr. Ewan just recently passed away, so I'd like to acknowledge both Professor Liggins and Dr. Ewan, and may they rest in peace. Thank you, everyone, and thank you very much for your attention.